Hi, this is Mrs. DeWitt, and these notes are about osmosis and tonicity. So we are going to start off with the definition of osmosis, which is the diffusion of free water across a membrane from high concentration to low concentration. For the rest of this video, I'm just going to be using HI for high and LO for low. And then also you will need to know that the brackets are an abbreviation for concentration, which is the amount of something in a certain area. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. So when we put cells in different environments, different solutions of different concentrations, they can actually cause a cell to gain or lose water through osmosis. And the three main types of these solutions are hypotonic, isotonic, and hypertonic. When we are looking at these prefixes, hypo, iso, and hyper, the prefix is actually referring to the amount of solute in the solution, not the amount of water. And you hopefully remember from chemistry or physical science in middle school that a solute is the minor part of a solution. And it is what is dissolved within the solvent. So a common solution we talk about is salt water. In salt water, the salt, the sodium chloride, NaCl, would be the solute, and the water would be the solvent, because the NaCl is dissolving in the water. So a hypotonic solution is a solution in which there is a low amount of solute. So when you think hypo, I want you to think low. And so we're going to say low concentration of solute. And if there's a low concentration of solute, that means there's going to be a high concentration of water. For example, distilled water. So distilled water has been distilled to remove as many solutes as possible. So that's one of the most hypotonic solutions there is. And we're going to start with that one. And we're going to start with this row with cells that have no cell walls, which are animal cells, which includes humans. All right, and now is where we're going to use some colors, pink and blue. We are going to use pink for a solute, and we're just going to use this triangle to represent a random solute that is in the solution. Here is an animal cell, very basic picture of it: cell membrane, nucleus, and endoplasmic reticulum. We are going to say that there's actually the same amount of water inside the cell as outside. So we got water, water, all around. We're going to draw just 5 molecules, for example. So we'll need to draw 5 on the inside too. Since this is a hypotonic solution, which means low, that means there is a low amount of solute in the solution surrounding the cell. So we're just going to draw 2 pink triangles because it's very low. And we're going to draw a bunch of pink triangles in the middle. where this represents the cytoplasm. And yeah, our cell actually does have quite a bit of solutes in it, any dissolved nutrients and gases and things. So we have a hypotonic solution because there are fewer solutes outside the cell than there are inside the cell. And so what we're going to do is osmosis, right? Is the diffusion of free water across the membrane from high to low. So the solute can't move, but water can. Since we have less solute on the outside and more solute on the inside, we actually technically have more free water on the outside, okay? Because it's not as diluted by all the solute. So water is actually going to move into the cell from high concentration to low concentration because we have a higher amount of water out here that's free than in the inside. And over time, since this doesn't have a cell wall, if we keep exposing the cell to more and more hypotonic solution, it is actually going to burst. Okay? So again, we had a high water concentration on the outside, low on the inside, so it moves from high to low, and then over time, it can actually burst. And there's a good picture in your textbook of a red blood cell bursting that you can check out. So, high water concentration outside the cell because there was low solute, it was mostly water, that causes the water to move into the cell where it's less concentrated, where there's more solute mixed into it. The way that I've been taught to remember this is hypo, like a hippo, because hippos are really big and the cell gets bigger because water is flooding into it. So water floods cell until it bursts. Another name for when cells burst is called when the cell lyses, just like Lysol, the ingredient that lyses cells, bacterial cells. So that is hypotonic with animal cells. Isotonic, iso means same, just like isomers, isotopes, okay? Iso means same. So isotonic solution, there's going to be the same amount of solute as solvent. Remember, the solvent is water. Okay. So here we have, let's just go with 5 again. Water molecules. And we'll put 5 on the inside. And let's just go ahead and have like 2 little guys on either side just for the sake of time. Okay. Since we have the exact same thing on the outside as on the inside, it's isotonic, that means the same amount of water is going to be going into the cell as is coming out of the cell. So there's still going to be movement, but it's called dynamic equilibrium. Dynamic means in motion. It's not static, which means to stay the same or not in motion. So dynamic equilibrium meaning water's still moving, but it's moving in at the same rate as it's moving out, which means there's no net movement. So remember, net means total. So since they're moving at the same rate in and out, there's no net movement. Water moves in and out at equal rates. So that's isotonic. A good example of when this is important is in the medical field, because if you need nutrients in the hospital and they give you an IV, a fluid, it needs to be isotonic. It needs to have the exact same amount of solutes and water in it as inside your body so that your cells don't expand and blow up, right? If we were to give you distilled water, that could happen. Another bad thing that could happen to your cells is if they're put in hypertonic solution. So someone who is hyper, it has too much sugar or too much energy. So you can think of hyper as a high hyper person, but high amount of solute, and a low amount of water. Okay, so here it's pretty much the opposite of our hypotonic solution. So we have a hypo low, hyper high. So here is my cell. We'll still do our same 5 waters. In blue, but this time it's going to be reverse, where we have tons of solute mixed in. So a good example of this would be like salt water or syrup, like really sugary water, okay, where there's a ton of solute mixed in with the water. So like the opposite of distilled water pretty much. And then we'll just put a few little solutes inside, okay? And in this case, now there is more free water, meaning it's not interacting with the solute on the inside than there is on the outside, okay? So since the concentration of solute is so high, some of this water is now going to move outside the cell. Boop, boop, boop. Okay, because we had a higher concentration now inside the cell and a lower concentration outside. So we always move from high to low. And we'll add that here too. Okay, so it doesn't always say inside to outside or outside to inside. It's always just from high to low, which could be on the inside or outside. Again, it depends on the type of solution surrounding the cell. And if too much water leaves the cell over time, instead of bursting over time, it'll shrink or shrivel up. And that actually, you might have experienced that if you go to the beach and you're playing in the ocean and you've accidentally swallowed some salt water. Your mouth might feel really dry and parched and your lips might be all wrinkled. And that's actually because you've exposed the cells in your mouth to salt water, which has a very high amount of salt, so that's high solute, and some of your water is leaving your cells. That's why you shouldn't drink salt water, because your cells will shrivel up, and that is not healthy. So again, water moves always from high to low, but this time the high side is on the inside of the cell. So in cell to outside of cell. Again, some examples could be salt water, syrup, which every good elf knows syrup is one of the main food groups. Okay. But this is all in animal cells. Hypo, low, hypo like a hippo because you swell. Iso is the same. You can think of when you're in a hospital, you need a high isotonic solution in your IV, and then hyper, really hyper, but that's going to cause your cells to shrink. Okay. All right, things change up a little bit when you get cell walls. We know that plants have cell walls, bacteria have cell walls, protists, like our friends, the algae, have cell walls, archaea have cell walls. So what happens with them is a little bit different because they have this rigid coat of cellulose or another polysaccharide like peptidoglycan or something. So we're going to add that in here, right? So we have the cell wall, and now we're adding in the membrane. So there's actually some space, teeny tiny bit, between the cell membrane and the cell wall. Okay, so the cell wall is actually on the outside, and the membrane is this line we just added. Okay, in a hypotonic solution for a plant or bacterium or whatnot, if we have more free water outside than inside, and we're going to just do 3 water molecules this time, just to speed it up. And we'll do 3. Okay, so while I was recording my video, I ran out of storage on my iPad, so that's how these other things just magically appeared. And so, just to recap, what I drew are 3 water molecules on the outside of the cell. We'll just go ahead and call this a plant cell. We drew 3 waters inside. However, this is a hypotonic solution, so there's low solute in the solution that's surrounding the cell. So I only drew 1 little picture of a solute here, whereas I drew 3 on the inside. So the concentration of water outside the cell relative to inside the cell is high. We have a high water concentration here, and it's considered low in here because this one has more dissolved stuff in it, more solute. So just like before, water will move from high to low always and come into the cell. Now, if we gave distilled water to an animal cell over time, it would just keep on letting more water in, and over time it would burst, and that's bad. But in a plant cell or any of these other cells with walls, this is actually a good thing, because as this fills and fills and fills and fills with water, it puts pressure from the cell membrane onto the cell wall. So we're going to draw some arrows here showing this pressure. And this is called turgor pressure. And as you know, it's very important for plants to stand upright and face their leaves towards the sun, because that's how they get energy. So this turgor pressure that pushes out on the cells is actually really helpful and makes the plant stand really tall, upright. So this is turgor pressure, and this is actually a healthy situation for a plant and an algae, unlike it being a healthy situation here. And so we have water moving into the cell here from high to low again, okay? And the cell wall and membrane exert pressure on each other. On each other. And again, just like I just said, this is called turgor pressure. Just try to say that out loud. Turgor pressure. And it causes the plant or algae to look turgid, which comes from this. And it actually prevents the cell from bursting, because there's this mutual pressure kind of back and forth here. So the cell wall prevents the cell from bursting, which is really good. And this is the healthy state of cells that have cell walls. So this is actually the ideal state, a hypotonic solution for our cells that have cell walls. Okay. So isotonic was the healthiest for our animal cells, but it's actually a little less healthy for our cells with cell walls like plants. So let's add that membrane in again. Okay. And in this case, we'll just do 3 waters on the inside. Outside. Boop, boop. 3 on the inside. And let's just do 2 little triangles to represent. So, and again, iso means the same, means equal, so there's equal amount. So just like before, we're at dynamic equilibrium, same amount of water in as is going out. So it's gonna go in and out at the same rate. So since there is the same concentration on both sides, water flows in and out at the same rate. But that means no turgor pressure. No turgor pressure. Means none of that nice pressure pushing back and forth on the cell wall and cell membrane. And this can make your leaves and stalks and all the other parts of a plant or algae go limp or cause them to wilt. And that's because there's not enough pressure to keep them upright. So if you have ever forgotten to water a plant, like I often do because I'm a terrible plant caretaker, and you see them start to wilt, this is why, because we don't have enough turgor pressure built up. They need a constant supply of water in the soil to keep this turgor pressure going. We don't want to overdo it because then that can cause root rot and microorganisms to grow on their roots, but we do want a healthy supply of water for turgor pressure, otherwise they wilt. Okay? But even worse than this would be giving a plant a hypertonic solution. So this is how actually sometimes you can even have your plants like die off if you give them way too much fertilizer, or if you've ever tried to give salt water to a freshwater plant, not going to go so well. And what actually happens, and we'll observe this under the microscope, see this membrane here? Just like the whole cell shrinks here, the membrane will shrink here. But the crazy part is, since the wall is stuck in place, the membrane just kind of like shrinks. So we're going to draw that in a second. We're going to kind of draw our water molecules kind of close so that you can see this. We just do water. We'll do 3 like we've been doing. But draw them close together because you'll see what happens in a second. And hyper again. So remember, hyper, lots of things. We're going to do lots and lots and lots of solute. Let's pretend that you are being silly and watering your plant with salt water. And we're just going to put one little solute guy in there. And what happens is now we have more water on the inside, less water on the outside. Hypertonic, meaning more solute than solvent surrounding our cell. So now water is going to move from a higher concentration inside the cell to a lower concentration outside the cell. So this water is going to leave. If too much water leaves this plant cell, the cell membrane, remember it was like really nice and close to the cell wall, actually shrinks. And it shrinks away from that cell wall. And all this space is created, and all of the cytoplasm and organelles are all squished together. Again, this is a stiff polysaccharide coat, so it doesn't move, but this all shrinks away. Very sad. So water still moves from high to low, from inside the cell to outside, and the cell membrane, okay, actually shrinks away from the cell wall. Sad face. This is called plasmolysis, and it is bad and will lead to the death of your plant unless it gets out of that hypertonic solution very quickly. All right, so to recap, the hardest thing you have to remember is that hypo, iso, and hyper refer to the solute and not the solvent. And so a hypotonic solution has a low amount of solute, which actually means a high amount of water. And the opposite is true of hyper, has a high amount of solute, so has a low amount of water. And think about that movement. There's some good pictures in textbook chapter 7 using red blood cells and plant cells as well. You are now going to try the rest of this packet on your own. So again, in these pictures, the little dots represent solute, not water. I want you to try this on your own, and then the key is going to be posted. And here's some other good pictures, like what I was talking about, like the ones in your book. So if you get stuck, look at chapter 7, and then try all of these as well. So this was all about osmosis and tonicity. Our next lab is all about this as well. We're going to be using different vegetables in different mystery solutions, and based on whether they shrink or puff up, you're going to make a prediction about the solution, whether it was hypo, iso, or hypertonic. Have a great rest of your day. Thanks for watching.